Hey guys, welcome to the Bio 285 Week 6 TA review session. This is the first review session of the two that we'll have this week, and we're gonna get right to the content. Chichi, wanna go ahead?
Yep. Okay, so the first topic we're going to start with is cell signals and their receptors. So we're going to do a deep dive into those. So in human cells, we obviously need a lot of signals to survive. That's really hard to keep them alive in the labs. Because and then cells receive a lot of signals at once, which tell them what to do. They don't just receive one at a time, multiples. I guess like the signal transduction. So it's like they get a lot of signals and then spread it out and tell us what to do. So signals are usually like ligands, which are small molecules, or they can also be proteins. And then these signals are received by receptors. They can be extracellular or intracellular. Extracellular, like usually in the outside, and then intracellular inside the cell. So, and then since we receive a number of signals, these signals can range from survival signals to growth signals to differentiation signals. And then if we don't receive those signals, the cells will die. And you can see the example of the apoptotic cell in one of the illustrations. Do you want to go back and forth, or is there any slide you want to do?
Oh, so I did them like sections, or if you prefer back and forth, I can do that.
I could just keep going.
Okay, cool.
So, like we said, the signals are obviously usually like ligands, which are small molecules. There's two different types: the cell surface receptors, and then there's intracellular receptors. So the hydrophobics are usually the intracellular receptors. They can pass through the membrane and bind to systolic receptors. Examples are like sex hormones or like thyroid hormones. Um, Nitric oxide is hydrophobic, so both nitrogen and oxygen, they can cancel each other out. So that's how they can get into the cell. And then the one we'll focus on mostly heavily is cell surface receptors. These can't pass through the membrane and need to bind. Sorry, hydrophils can't pass through the membrane, so they have to bind to the cell surface receptors.
Yeah, and also I was gonna mention that the nitric oxide is actually considered like a hydrophobic signal. So hydrophobic signals are able to pass through the cell membrane. And so it's actually hydrophobic, even though it has two electronegative atoms, but because they're highly electronegative, they're basically the charges kind of cancel each other out. So um, that's why it's considered the compound nitric oxide is considered hydrophobic. Just thought I'd add that.
Okay, so two outcomes of cell signaling. Can either cause a change in protein function, or it can cause a change in protein levels, which is done through gene expression. So both of these processes are very highly regulated. It is, although, faster to change or alter an existing protein function. They're not mutually exclusive, so they can both happen as a result of the signal transduction pathway. So multiple receptors, multiple receptors can receive the same signal, but they have different outcomes from that. So depending on the cell type, the signal may affect it differently. So different cells express different proteins. For example, the heart muscle, as you can see in the illustration. So it binds acetylcholine, and then that causes a decreased rate and force of contraction, versus the same signal will bind the receptor protein in the salivary gland cell, and then that causes secretion of from the salivary glands. And then the skeletal muscle, it will just cause contraction. And then, so these heart rate increases, digestion is slow, like saliva accumulates and stuff like that. And then, so in receptors, the extracellular, their signals are usually large and hydrophilic, like we talked about before. And then for the intracellular receptors, the signals are usually hydrophobic, so they can penetrate through the membrane and get into the cell. So then the same signal receptor can produce different outcomes, like we talked about before.
I just wanted to stop real quick. Did anyone have any questions on anything that was said so far?
No, they said that.
Nope. Okay, perfect. Thank you, Haley. We'll continue. Now I'm going to jump in and talk about signal amplification versus crosstalk. So signal amplification is commonly used in some pathways, and it's basically when you have a signal that's able to be amplified if there's proteins or enzymes in the pathway, or these components that represent like multiple steps within a transduction cascade. So you guys can see in the image that you have like a signal binding to the receptor, which causes components of the pathway to become active. Then one component activates another component. And then you see this specific component can activate multiple components at a time. And so that kind of causes the whole cascading effect to occur. And I included a video about signal amplification, just so you guys can distinguish it between signal amplification. So I'll play that.
Receptor proteins interact with signal molecules at the surface of the cell. In most cases, the signals are relayed to the cytoplasm or the nucleus by second messengers, which influence the activity of one or more enzymes or genes inside the cell. However, most signaling molecules are found in such low concentrations that their effects in the cytoplasm would be minimal unless the signal were amplified. Therefore, most enzyme-linked and G-protein-linked receptors use a chain of other protein messengers to amplify the signal as it is being relayed. In the case of a protein kinase cascade, one cell surface receptor activates many G-protein molecules. Each G-protein activates many adenylyl cyclases. Each cyclic AMP, in turn, will activate protein kinases, which then activate several molecules of a specific enzyme. The end result is rapid production of high levels of the final product.
Cool, so that was just a video about signal amplification. I thought I'd incorporate in there just so you guys can see the, just so you guys can see the difference between signal amplification versus crosstalk, which I'm about to talk about. So yeah, so that's signal amplification. And sorry, just a second.
Oh, sorry.
It's okay. So now I'm going to talk about crosstalk. So crosstalk basically happens when you have components of one pathway directly affecting another. And it basically kind of means like when one protein for one pathway can talk or like influence protein from another pathway by changing the function or behavior of that protein in some way. And this happens in cells all the time. And you guys can see, I don't know if I have the clicker. Oh. You guys can see like in this example here, you have the growth factor signaling pathway and this hormone transmitter pathway, some of the proteins from a different pathway communicating with RAS and causing either activation of RAS. And you can see in other examples, the arrows mean that things are being like activated. And then if there's like, I can't see where it is, but some of the areas you'll see that there's like kind of like a T, like an arrow, but without like the arrowhead. So kind of like a T, which means that like it's inhibiting or being blocked. So like this ARF component is blocking NDMM2 in the nucleus. So yeah, so you can see that it's normal for cells to have pathways that like multiple pathways, and that pathways kind of relay messages to each other and participate in this crosstalk. Like they don't just have like a singular pathway that has events that occur by itself. Like they're always communicating with each other. So an example of this is like when you have a cell that receives a cell death signal to undergo programmed cell death, then that means like the cell death is occurring. So that means the cell division signal shouldn't be occurring at the same time. Because if a cell is going to experience apoptosis or cell death, then that would mean that the cell survival signal, it wouldn't make sense for the cell to also be getting the signal for survival. So that's how pathways sort of interact each other. So maybe the cell death pathway could suppress the survival signaling. And so, yeah, that's crosstalk. And the next slide, I just thought I would give like an analogy about crosstalk, just so to see if it like made a bit more sense. So here I have like two groups of people. There's group A and there's group B. And in um, group A, there's like 5 people in the group, and they're trying to like relay a message to each other. And so, like not thinking about biology or anything, this is just sort of like a real life analogy. Kind of like if you ever played like I think the game's called like Chinese whispers, where you like whisper something to the next person until the last person kind of gets the message that was relayed across from the first person. So basically in group A, the message that the first person is trying to send is that today is Wednesday. So the first person tells the second person the message that today's Wednesday, and so on and so forth until the last person receives the message that it's Wednesday. And so that means that the message was transduced across the entire group. And so that's essentially the same idea with group B, except group B has a different message. So group B's message is it is March. So just like group A, group B would say, the first person would tell the second person that today's March, and then they would pass on that message until the last person received the message that today's March. And so you can kind of see the analogy with the protein pathways, because the groups represent pathways and each individual person could represent a component, and the message that they're trying to pass on to each other would represent your signal. And so with this example, I just decided to put an example of what would happen if the signal gets interrupted somehow, or if the message doesn't make it through. So in this example up here, you have group A, which had the message today's Wednesday. But if you look at the group, the third person is actually asleep. So the message doesn't get relayed all the way through to the last person. So the first person tells the second person that today's Wednesday, but because this third person is asleep, or this component is disrupted or deactivated, then that means that the message won't be transduced throughout the whole group. So the message doesn't get received, and so the message is unable to be transduced all the way through the pathway. So hence the X here. And so with group B, everything is the same. So their message that it is March is still able to be like relayed across the group or across the pathway, if you want to think about it that way. And so what I was trying to show down here is that in order to help group A's message get transduced all the way to through the pathway, a person in group B could actually run over to someone in group A and shake them awake, which would cause them to become active, and then they would be able to relay their message. Group A would be able to relay the message that today's Wednesday, and the signal would be received or transduced all the way through. And while the person from group B is doing that, the group B signal or message, which is it is March, is able to be transduced also. So hopefully that makes a bit more sense, but I was just trying to put an example of crosstalk in by using people and the groups. But you can see how like one group can influence another group, and it's able to help them get the message across the entire group, which would represent a pathway. So can I get like a thumbs up or something in the chat to see if that made sense to people about what crosstalk is? Let me see. Great pictures. Okay, perfect. Thanks, Des. Cool. Yeah, so that's basically crosstalk, and glad you guys understand. Cool. So yeah, so this is just a cell signal summary of stuff that I said and stuff that Chi said. In general, signals are typically small molecules or proteins that bind to intracellular receptors or extracellular receptors. The same signal binding to this exact same receptor on different cells can have different responses. And cells can receive a wide variety of signals from survival, growth, division, differentiation. And if cells don't receive like multiple signals or survival signals that they need, then the cells will die. And we already discussed signal amplification, but it's just when a signal becomes amplified. And crosstalk we just discussed, which is just interaction between elements of different signaling pathways, so that a component of one pathway can affect a component of a different pathway. And yeah, that's just a nice summary slide of everything that we covered so far. So any questions so far?
Okay.
I'm gonna take that as a no. And Chichi, you can go ahead and do the next part.
Now we're going to be talking about kinases and RTKs. So let's just do a quick overview of protein kinases. There's a lot of kinases. Some we already talked about, like tyrosine kinases, and then there's obviously serine and threonine, just from the these are elements that you would see in like a polypeptide. So then tyrosine, serine, and threonine are groups all share hydroxyl groups. So kinases are proteins and sometimes enzymes. They usually cleave off phosphate groups from ATP and attach it to a substrate, specifically the oxygen in the substrate. This is important because kinases don't just phosphorylate things. They're involved in the process of cleaving the phosphates and attaching them to other substrates, which is usually like another protein. Yeah, this is kind of what we talked about. This is like a visual example of coming from dephosphorylate protein to a phosphorylated protein. So it's like the kinase cleaves a phosphate and changes ATP to ADP, and now it's phosphorylated. Then receptor tyrosine kinases. They're family receptors for survival, cell division, cell differentiation, and insulin signals. These types have a receptor domain, as you can see in the illustration. They also have a transmembrane domain and an intracellular portion of the protein that includes a kinase domain. Again, a kinase is an enzyme that catalyzes phosphorylation. So as you can see in the illustration, the signal molecule is in form of a dimer, and then it binds at the receptors, and then it phosphorylates the tyrosine and it activates the receptor tyrosine kinases. So general mechanisms of RTK signaling: the signal binds, which is called it's in front of a dimer, so it's dimerizes or dimerization. And then the intracellular kinase domains, which are in the illustration, then they become active due to the conformational shift. So the two come together and then it causes a shift. and then that causes a cross-phosphorylation of tyrosines. So the tyrosines in the illustrations, they cross-phosphorylate each other. And then the RTK autophosphorylation creates binding sites for intracellular proteins, or the intracellular signaling proteins. So it's like, as you can see in the others to the right side of the illustration, there's a lot of binding sites for things to be binding into. So signaling binds to two inactive RTKs, and then this brings them close together. So this is what the dimerization is about in like the illustration we showed on the previous page, with the two of them coming together. So the two proteins come together to form a functional unit, and then this causes a conformational shift in the kinase domains inside the RTK, so in the intracellular portion, and then this activates kinase activity. So then the conformational shift transmits the signal information from the outside to the inside of the cell. And then activation of kinase activity in the two RTKs cause them to phosphorylate each other. This is called sorry, I mess this up a lot. Cross-phosphorylation or autophosphorylation. So the RTKs have cross-phosphorylation of tyrosines, which resides in the intracellular portion of the RTK. We'll be focusing on 4 pathways this week: the RTK, RAS, MAPK pathway. These types of pathways are used for cell division and cell differentiation. The next pathway is the survival pathway, which is the RTK, PI3 kinase, and AKT pathway. They are for cell survival signaling in multicellular organisms. It's kind of like the ones in our bodies. And then we have the JAK-STAT pathways. They're used to signal for cytokine signaling, so like in the immune system. They're also used for cell division, and for example, we'll use the milk protein production during breastfeeding. And then the last pathway is the GPCR pathways. These are the most common type of receptors in the human genome, with around 800 different versions. So they're G protein coupled receptors. And then we'll be covering the RTK, RAS, MAPK pathway for today, and then the rest will be talked about in a review session on Friday. Take it away, Michelle. Oh, does anyone have any questions?
I was about to say the same thing. If anyone had questions. No? All right. We'll keep going. All right, now I'm gonna dive into the first of the 4 pathways we're gonna talk about this week. This one is the RTK RAS MAPK pathway. Okay. And before I start, I just decided for like organization's sake and just for like easier to comprehend, instead of having like a huge picture of the pathway, I broke it down into smaller steps for you guys to see. And then at the end, I'll have a picture of the entire pathway, and just like on a blank page, just so you guys can like sort of practice walking through like each of the events that happens. Just so you guys like understand how I have the slide set up. Cool. So first thing that happens in the RAS MAPK RTK pathway, the receptor tyrosine kinases will begin the pathway, and you'll probably notice this is a common theme for most of the pathways. So first thing that happens is the signal comes along in the form of a dimer, and so that will bind to the RTKs, and that will cause dimerization of the RTKs, meaning that the RTKs are able to come closer together in proximity. And once the dimerization occurs, then this will cause the intracellular kinase domains to become active. And this is because when they're in close proximity to each other and it's bound to the signaling molecule binding to the RTK causes a conformational shift, or in other words, causes like a shape shifting change within the intracellular portion of the RTK. And so that's what causes the intracellular kinase domains of RTK to become active. And now once you have the active RTKs, they're able to what we call cross-phosphorylate each other. And so the RTKs are close enough to be able to cross-phosphorylate each other. And that occurs on the tyrosines, on their tyrosine kinase domains. And once the RTKs are active and they cross-phosphorylate each other, they're able to use these phosphorylated sites as binding sites for intracellular signaling proteins. And these binding sites actually have a special name. They're called SH2 domains. And anytime you see a phosphorylated tyrosine in a pathway, just know that there's usually that component has an SH2 domain, which enables like other components, which are most of the time usually their proteins, to bind to that area. So again, that's just called an SH2 domain, and it's just an area that enables it's an area that enables like a protein to bind that has a phosphorylated tyrosine. Cool. Does anyone have any questions on that part so far? I'm going to take it as no, and I'm going to continue. So once you have the activated RTK, the next part of the pathway can happen. And so what will happen next is the adaptor protein is able to bind onto the SH2 domain, which is this phosphorylated tyrosine site on the active RTK. And once the adaptor protein, which is also known as GRAB2, binds to the phosphorylated tyrosine, it is able to recruit the RAS activating protein, which is, I think, called SOS or SOS. And I think also you can refer to it as RAS-GEF, but I just call it the RAS activating protein because that's what the diagram says. But SOS is also fine. So what will happen next is, once the RTK becomes active, the adaptive protein binds to the SH2 domain. And then once that occurs, the adaptive protein can recruit the RAS activating protein. And once the RAS activating protein is recruited, then it is able to help the RAS protein in swapping the GDP for GTP. Or the way you could describe this is that basically the RAS activating protein causes RAS to have a lower affinity for GDP. So what will happen is RAS will dissociate, or the GDP will fall out of RAS's binding pocket. And so because there's more GTP that is abundant in the cell, then the GTP is able to diffuse into the binding pocket of RAS, which you can see here. And once GTP is bound to RAS, it is now in its activated state. And so that's basically what this diagram is showing. So the RAS activating protein swaps out the GDP for GTP, and that's what allows the RAS protein to become active. And yeah, another thing to mention is in the diagram, RAS is actually anchored to the cell membrane. Just something to know. So that when the RAS activating protein is able to like help RAS become active, it's the GDP that comes out of its binding pocket, and like RAS is kind of in a fixed position. And so the RAS activating protein just has to be in close proximity to RAS. Yeah, and so that's that section of the pathway, which is like the beginning half. Does anyone have any questions on anything I described so far? You can drop something in the chat or unmute. Let me see. No questions. Okay. Cool. I'll just keep going. So that's the like half of the pathway. And so now I'm going to continue to talk about what happens after RAS, which we saw here, becomes active. So once your RAS protein is active, it's able to continue on in the pathway. And so what it will do is it will interact with MAPK3. So, and MAPK3 is also called MAP kinase, but since it's kind of redundant to say the kinase 3 times, I just abbreviate as MAPK3. And I think you guys are allowed to do that as well. Just make sure that the first time you do it that you say that it stands for MAP kinase, and then you can just sort of like write it like that. So anyway, so the activated RAS will interact with MAPK3, and it's important to distinguish here that RAS is not a kinase, so it does not phosphorylate MAPK3. It just sort of interacts with it in a way that causes MAPK3 to become activated. Um, and so once the MAPK3 um, is touched by the RAS protein, then it's able to continue on and activate MAPK2. And because MAPK3 is a kinase, it will phosphorylate MAPK2 at two different sites, at two different positions. And then what will happen next is MAPK2 will phosphorylate MAPK1 at two different positions. And then once these phosphorylation events happen, it causes like activation. So like the kinase will phosphorylate another kinase and cause it to become active and phosphorylate another kinase. And so then eventually you have MAPK1. And once MAPK1 is phosphorylated by the MAPK2, it's able to go and phosphorylate other proteins or phosphorylate transcription regulators. So yeah, so the MAPK1 can result in changes in protein activity or changes in gene expression. And so that's like the bottom half of the pathway. Does anyone have any questions so far on what I described? Does it make sense to people? It's just kind of the idea of like kinase cascade and how you have MAPK3 activating MAPK2, MAPK2 phosphorylates MAPK1, and then MAPK1 can phosphorylate like downstream proteins or transcription regulators. And RAS does not phosphorylate MAPK3 because it's not a kinase, so it just causes MAPK3 to become, it just sort of interacts with MAPK3 in a way that makes it active. Let me check the chat. Nope, no questions. Okay, and it makes sense. Cool. Okay, so that's pretty much the gist of the pathway. And so as you can see, I kind of broke it down into like two halves, but this is the entire pathway. And I strongly suggest that you guys kind of walk through the pathway yourselves, like when you're studying or when you're looking at the pathways, because a good way to tackle these kind of questions is to understand how the pathway works normally. So kind of the sequential order of things, like what activates what and like the different events that happens. And so that'll prepare you to answer questions like what would happen if you deactivated RAS protein, or what would happen if you had overactive MAPK1, and those two sorts of questions, those problem solving questions are things that Laura will ask you guys. So yeah, it's just taking the concepts of understanding how the pathway works normally to be able to answer questions that deal with what would happen if you had like mutations or abnormal situations. So yeah, strongly encourage you guys to like take the slides like these that are empty and just sort of practice these yourselves, like talk to a cat or a goldfish, or maybe someone who hasn't taken a class, or your parents, and see if you can like kind of explain what's going on, because being able to explain it to someone else shows that like you have an understanding of it. And yeah. And so this slide here is just sort of everything that I just said before is in this slide here. It might be a little bit of a clearer description. I just know that people are different learners. So some people like the pathway broken up into steps and explained that way. And other people like to have a whole list of everything that happens all at once, and then other people like to see visually like the entire diagram. So I just thought I would include the steps that happens. I don't feel it's necessary to go through it all again. This is just something you guys can read in your own time, or if you want to quiz yourself on like the sequential events that happen. But yeah, this is just a summary of everything I just said. So I'm just going to go ahead and skip this part, unless anyone has any questions, which I don't think so. So keep going. And yeah, that's pretty much it for today. Um, we went through the pathway, one of the 4 pathways today, and just like cell signaling characteristics and things like that. And so the next review session on Friday, we're going to cover the other 3 pathways. We just figured we do 1 pathway today just so that you guys don't feel overwhelmed about it because it's a lot of stuff. We know that. And yeah, that's pretty much it for today's review session. And we'll stick around if you guys have questions. But thanks for coming.
Thank you.